Okay. We know that pesticides are a very important group of environmental pollutants used in intensive agriculture for protection against disease and pests. However, pesticide residues, or the improper handling of this compound can result that pesticide residues can be found in soil and water in the form of pesticide wastewater and has obsolete pesticide. Now, considering the toxicity and danger of this compound, decontamination of polluted environment or pesticide wastewater are essential. Develop. Okay. Now, finding efficient treatment system for pesticide has been a preoccupation in the area of science and engineering, and also for our research group. Now, a pesticide group that has received considerable attention are organophosphate, because these compounds are the most widely used pesticide, are inhibitor of enzyme acetylcholinesterase, present and moderate to high toxicity, are carcinogenic and mutagenic compound, and are contaminants of soil and water. Diazinon is an insecticide widely used in agriculture, livestock, and as well as in dogs and cats, in house. Now, a promising method for pesticide detoxification is by using microorganisms. Actinobacteria or actinomyces are bacteria recognized in medicine for the antibiotics production, in agriculture for production of compounds with biopesticide and biofertilizer properties, and now in environmental science for the ability to degrade various contaminant compounds, including different chemical class of pesticide. And moreover, this actinobacteria present physiological and biochemical characteristics, are bacteria resistant to adverse environmental conditions, so are considered adequate to be used in bioremediation system. Yeah? No? Okay. Now, the use of microbial consortium could support enhanced pesticide degradation, because different metabolic pathways can act. So, therefore, the objective of this study is to select an actinobacteria consortium to enhance the diazinon degradation. In this study, organophosphorus degrading actinobacteria identified as Streptomyces strain mentioned there were used, and the standard of diazinon and the main metabolite IMHP. The study with single bacteria, spores were pre-incubated in liquid culture medium to form in a rich medium for biomass production. Yeah. Then biomass was used to evaluate diazinon removal at 50 milligram per liter concentration as only carbon source. After incubation, we evaluate biomass, protein, and diazinon, and IMHP. After that, we perform the strain selection for the next study with mixed culture. Yeah, and an antagonism assay was performed between the different strains, and then we evaluate the diazinon removal as only carbon source. Yeah, different mixed culture were evaluated. And this, diazinon removal by single actinobacteria is shown in this figure, and it can be seen that with single actinobacteria, 10 to 30% of diazinon was removed. Yeah. The highest diazinon removal was observed by the strain for three strains, yeah, AC9, GA11, and EASP13. Respect to biomass growth, 6 of the 8 studied actinobacteria were able to grow with diazinon, which could be used as carbon and energy source. Okay. In this figure, we can observe the protein profile evaluate for each strain. Yeah, and the protein profile of the cell-free extract showed increased intensity and more protein bands in the line that represent the strain cultivated with diazinon. So, diazinon induces enzymes in the actinobacteria. Exceptions were observed for AC7 and EASP4 that show similar profile without diazinon and with diazinon. Yeah. The molecular weights were between 34 to 78 kilodalton that could correspond to a phosphotriesterase that can degrade organophosphorus compound. But another study are necessary to clarify this response. Now, with this result, the strains that were selected were AC5, 6, 9, GA11, and EASP30 for the next study with mixed culture. Okay? The antagonism assay showed that the strain AC6 was the only one that showed a slight growth decrease when fasted with a strain AC5, but in general, no inhibitory effect was observed between the strain. Okay? The next study we performed 25 different combinations of mixed culture, yeah, and with the final aim of to select a mixed culture with higher diazinon removal. Yeah? We can observe that the use of mixed culture increased the diazinon removal. Yeah? And on average, the combination of two strains, diazinon was removed in about 34%. 34%. 48% was observed for the culture formed by 3 strains. Yeah, and the combination of 4 strains showed an average degradation of 58, and the combination of 5 strains showed 28% of diazinon removal. According with the results, the selected actinobacteria combination were streptomyces, this mixed culture, formed by the strain AC5, 9, GA11, and EASP3. So the consortium or mixed culture was formed for 4 strains. Respect to the IMH production by mixed culture of actinobacteria, we can observe that the concentration of IMHP were between 0.16 to 4 milligrams per liter, and the highest IMHP production was mainly observed when a combination of 3, 4, or 5 strains were used. Now, IMHP is reported to be the major diazinon metabolite found in compost, soil, and water. Yeah, and its characteristic has a less toxic compound compared with than diazinon. After selection of mixed culture, we evaluate the degradation, diazinon degradation at different time, and the result showed that approximately 50% of diazinon was removed in the first 24 hours, and biomass duplication was observed. So diazinon was removed and biomass increased. And now, respect to IMHP, the metabolite of diazinon appear at 3 milligrams per liter concentration and decrease over time. At the final of a site, no residues were detected. Yeah? Okay. As conclusion, mixed culture of streptomyces increased the diazinon removal. Diazinon degrading enzymes are expressed by some strain of streptomyces. Diazinon is used as solid carbon source for growth, but studied streptomyces, and formation of the diazinon metabolite was observed. So I think that this culture could be used in a treatment system for diazinon removal, but the study continues. Okay. The study is supported by this financial support, and thank you. Sorry. 50 milligrams per liter. Yes, 50.
You reach to 20% or 30% only?
Only a single strain removed 30%, but mixed culture close to 90% at the same concentration. Only streptomyces. Only.
Do your strains or the consortium grow on IMHP?
Sorry?
Do your promising strains, individual strains, or the consortium grow on IMHP as a carbon source? The product of diazinon degradation?
No.
The product of diazinon degradation, namely IMHP, if you provide, do the strains grow on IMHP?
No.
No?
No. Now, and at this moment, I'm working in IMHP degradation. Yeah, I'm working with organophosphate pesticide chlorpyrifos and diazinon. Yeah, so many studies are doing at the moment. Yeah, but
no. But if they are growing on diazinon, they are breaking it down and reusing it as a carbon source, they should also grow on the product of degradation, namely IMHP. Should they not? We'll discuss it.
coffee.
Yeah, sure. We need another coffee.
Sorry.